Dear students, friends, viewers all over the world, my name is Dr. Byproduct, and we are here to present one of our lecture series titled Food Analysis. Food Analysis. You can see it boldly inscribed. Food, as we all know, is an important substance needed by man and animal for sustainability, for growth, and survival. Across the globe, people eat food in different formulations, either in liquid or in solid, as recommended and regulated by every nationals. Because of the importance of this substance called food, a lot of reports have been documented on their spoilage, and the dominant living organisms that cause this spoilage are microorganisms. Now, the presence of these microorganisms lead to the deterioration of these food substances, and the end result is that it leads to disease condition. So, as a result of this, it's important we analyze the microbial safety and quality of this food to see their status, microbial status, and to know whether recommendations can be done where necessary. Several techniques or methods or assays have been developed to analyze these food substances. In this lecture here, we are going to look at some of them, starting with membrane filtration, standard plate count, molecular analysis, metabolic byproducts, selected group analysis, selected pathogen analysis, direct microscopic count, most variable number technique, and so this is where we're going to limit our lecture for today. So for more emphasis, I want to start with standard plate count. In the standard plate count, the food sample is prepared, the suspension of it is prepared, and then series of dilutions are made depending on the food or factor of dilution of choice. Mostly 10 is being used. Then a selected medium or medium of interest is prepared according to the manufacturer's instruction. So they are sterilized, poured, and then after the dilution, an adequate quantity, depending on the technique, either standard spray plate or pour plate, is being adopted and then inoculated in this plate. The plates are incubated for bacteria at 37 for 18 to 24 hours, for fungi at 25 for 72 to 96 hours. Thereafter, colonies that emerge on the surface of the plate, which are dotted outputs, are counted, and then results are expressed in CFU. CFU is an acronym that stands for colony forming units. Colony forming unit per gram. If the food sample is solid, or per ml if the food sample is liquid. So this is how results of SPC are being expressed and documented. Okay, then the next one is membrane filtration with the acronym MF. In the membrane filtration here, the food sample is prepared. Mostly because of this technique, liquid food samples are used. Here, an adequate quantity, preferably 100 ml, is being filtered under this filtration system using mechanical vacuum pump, and then for a particular period of time, at the end, the membrane filter, the filter membrane is here, usually about 0.45 micron. Okay, micron is being used. So after the filtration, it is aseptically taken and dropped in a selective medium of choice. And then at the end, colonies are counted. Colonies are counted. And these colonies are reported after incubation as what? Colonies. We report them as what? Colonies per 100 mil. Remember, we use 100 mil to carry out the filtration. We use 100 mil to carry out the filtration. So, colonies per 100 mil. This is how it is being reported. The next one is the most probable number technique. The most probable number technique. The most probable number, which is the acronym MPM. This most probable number technique is carried out in 3 stages. 1, the presumptive; 2, the confirmatory; 3, the completed. So the presumptive is carried out either using 3-tube technique or 5-tube technique. When we say 3-tube, we mean 3 in 3 series, or 5-tube technique, 5 tubes in 3 series, 5 in 3 series. So here's where the choice is yours to adopt. Okay, so if we're using, for example, the 3-tube technique here, suitable liquid medium is used, like lactose broth, laurel broth, microscopic broth, and so on. So you prepare it in double strength and what? Single strength. You sterilize and then bring it to cool. Also, the food sample, either in solid or liquid, is prepared. The suspension of it is prepared, and aliquot of this is inoculated into the what? The medium. Now, in different dilution, for instance, 10, 1, and maybe 0.1. Yes, 10, 1, 0.1 into the 3 different cells. So here now, the target here is to check for the tubes that have positive gas formation. Gas formation is our target here. The gas formation is our what? Target here. So the gas formation is our target. So we look for the tubes. So at the end we record. For instance, we say 3, 2, 1. If I have this, it shows me that the 3 tubes, the first 3 tubes are positive. The second set, only 2 tubes are positive. The third set, only 1 is positive. So the result of this are being recorded and documented and carried to, and then compare from a table called MPN table. So from that MPN table, it will give you a particular cell number. It will give you the probable number of coliform that are present in that food sample. Okay, so the confirmatory test now, for the tubes that record positive here, we carry out confirmatory tests where we can just get from the tube a streak in a selective medium like EMB, eosine methylene blue, and we check for coliform, particularly E. coli. For the E. coli, it has metallic green sheen colonies. When the appearance of that shows that it's confirmed that it's coliform, particularly what? E. coli. And then from there, we proceed to the 3rd stage, which is the completed. In the completed, we can redo what we did initially in the presumptive, and then we can repeat that, and then repeat it, and then do gram staining. We can do spore stain to know that it is spore positive or negative, or gram positive or gram negative. Okay, coliform usually are gram negative. Coliform are spore negative. They are non-spore formers, and so with this we complete it. Okay, so we call it most probable number because we want to look the most probable number of microorganisms, particularly bacteria, in this case coliform, that are present in that food preparation. Okay, now the next step is the direct microscopic count, here also known as DMC. Okay, now this method, it is usually carried out only for liquid food preparation like dairy products, milks, and the rest of them. So carry it out. So in this case, the food sample is prepared and then stained using a suitable stain like Japan blue and so on and so forth. So we stain it, and then an aliquot is aspirated and dropped into a microscopic counting chamber, like what we call counting chambers. Okay, like what we call counting chambers. We drop it in that counting chamber and then view under the microscope. From that word microscopic, Okay, that is why we are using microscope. So this counting chamber, we drop the sample inside, then view under the microscope. So we count cells within these squares. We see squares under the microscope. So cells within these squares are counted and recorded. So each counting chamber has a factor called microscopic factor. So look at the counting chamber. There is an inscription of MF. Microscopic factor has an acronym MF inscribed in that counting chamber. Look at the factor and the easy to. Because the number of cells you can, for instance, you counted 250 cells. So you multiply by what? The microscopic factor the manufacturer has given you. That will give you the cells per mil or the cells per gram. Per gram if it's solid, or per mil, not per gram, per mil, because this is purely for liquid samples. Okay, per mil. So this is how this DMC is being carried out. So coming to the selected pathogen, S. aureus, B. bacillus cereus, and so on. So several pathogens exist: Ewelium, Vibrio, Clostridium, and several of them, depending on the choice you want to use. So any of these selected pathogens can be isolated and identified. Okay, using a selective media like E. coli, we can use chromocort agar, EMB, eosin methylene agar. For the Staph aureus, we can use mannitol salt agar. Then for the bacillus cereus, we use bacillus cereus selective medium to isolate them in the laboratory. Okay. The next one is the selected group. Under the selected group, we can also study the groups of food microbes, like coliforms. Okay. So we can use the chromocort coliform agar to get the group as well. Okay. The Enterobacteriaceae. Okay, we can use endo agar to get the Enterobacteriaceae. The Clostridia, we can use Clostridium selective medium to get them. And then this Staphylococci. So here is a group. So we can also use the mannitol salt agar to get the group listing. The next step is the metabolic byproduct. Here, byproducts, once microorganisms carry out metabolism, they have an end product. That end product, one of the end products is toxins. So these toxins mostly can come from bacteria, fungi, algae, and this. So the one that come from fungi are mycotoxins. The one that come from bacteria are what? Bacteria toxins. These toxins can be produced externally, which we call exotoxin, or internally, which we call what? Endotoxin. So usually in the lab, we can use the food material can be prepared. Some separate analytical technique can be deployed, like HPLC, high performance liquid chromatography, can be used to detect the presence of toxins quantitatively and qualitatively. GCMS, gas chromatography mass spectroscopy, GCMS, gas chromatography mass spectroscopy can also be what? Adopted. Thin layer chromatography can be adopted. TLC, okay? Affinity chromatography too. Affinity chromatography can be what? Adopted, and then be used to detect the presence of these toxins in these samples. Then the next one is the molecular analysis. The molecular analysis is either we do community study or specific study. The community study is what we call, where we deploy the technique of metagenomics. The metagenomic primer is being designed and then used to get the community, the structural and functional food microbes present in that food preparation. For the specific one, we can use the PCR. PCR is an acronym that stands for polymerase chain reaction. FISH is an acronym also that stands for fluorescent in situ hybridization. Here, an oligoprimer is being designed and then used to detect the presence, or qualitatively or quantitatively as well, the presence of food pathogens. So these techniques are the ones we dwell on these lectures. So as important as they are, also there are several standards that have been adopted in Europe, America, and developing countries. A lot of manuals are being established in those countries. Okay, that manual is what we call pharmacopeia. 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 Pharmacopeia, or what we call compendium. It's a compendium, a big manual, so big. Each countries have their own pharmacopeia, and there are standards in those pharmacopeia depending on the food formulation, either in liquid or solid or semi-solid, depending on the food. For instance, now some of you are we know that you are going to work or you are already working, maybe earlier. So those companies or institutes where such food can be exported. Like for instance, the interest is to export food maybe to America. You see, as you are running the analysis, you use the manual AP. American Pharmacopeia is called AP. British Pharmacopeia is called BP. Chinese Pharmacopeia is called CP. Indian Pharmacopeia is called CP. So if you are exporting, for instance, to America, you use AP manual of Pharmacopeia so that in each you see the standard. For instance, now I have a food product, a grain, for instance, and I want to do SPC. I carry that SPC on that food, and in the Pharmacopeia, the result is 500. The limit is what? 500 to 600. The limit in the Pharmacopeia is what? 500 to what? 600. 600 CFU per gram on the series. So in this case, if I obtain colonies below 600 colonies, so at this point, colonies, if my result is below, the food will be what? Accepted and then to be exported to America. If it is within, it is marginally accepted, okay, and can still be sent. But if it is above this range, I should what? Reject, and possibly maybe there is a procedure error and I can repeat the procedure. And if captured again, so I would recommend that the food should be highly what? Treated before exporting. So this is how to interpret results based on pharmacopoeias. So each nation have their own pharmacopoeia. Even Europe, European Pharmacopeia is EP. EP is European Pharmacopeia. So as that. So food analysis is important because of the food is important because of its importance to us because of the survival, and this and some other analyses are important to be carried out. Okay, it's important we carry out this analysis in order to guarantee the quality, in order to guarantee the safety of this food. Because if analyses are not well done, or if they are done and precautions are not taken, the final consumers, which is you and I, can be victims. Can have food poisoning, and if not well managed, can lead to death. So, for the sake of humanity, it's important to carry out microbial analysis under the microbiological aspect. My friends, viewers, I hope you enjoy throughout today's lecture series. And if you enjoy it, we appeal that you do what? You subscribe, you like, okay, you share, and then you can also what? Comment where necessary. Maybe an aspect of where I had initially discussed is not clear, so you can comment, and we shall respond at the appropriate time. We will come and attend to you. Okay? So thank you for listening and giving me your audience. And do stay safe and remain blessed. Thank you. Bye.